right? Your RNA is the translation into whatever your language is. So for my case, it would be English. So that way I know how to put together my bookcase. And all the little pieces are the proteins that I'm going to need to put together to build my bookcase. So I
Everyone, all
now have a
backgrounds, all
functional thing
ages,
in
all
my house.
genders, and all walks of life deserve an education. Come be one
Alrighty,
of
well, Alicia, thank you so very much for joining us today, guys. Very, very special guest. This might be one of my very best friends for a long, long time. Dr. Alicia Mortimer. She is a researcher that I've known since my days as an undergrad. She is a professor. She's an award-winning researcher. Experimental gerontology, remember, is the her first award-winning paper. She does scientific presentations, scientific outreach, and just far so much. We're going to be chatting with her today about her journey in the sciences, as well as the research that she does. Alicia, welcome on in.
Oh, thank you for having me, Bullet. I've been really looking forward to our chat today.
Alicia, we're really excited to have you. The community has been even asking like, "Oh, can it just be a?" Because she like sprinkle in anecdotes about silly stories about the how y'all were together. I'm like, no, it's serious science. But maybe we'll sprinkle in a couple of silly stories too. Would you mind giving an intro to everybody?
Sure. So, hi everyone. I'm Dr. Alicia Varales Mortimer. My research focuses on understanding the genetic and environmental factors that contribute to aging and age-related diseases, and I use fruit flies as my model system. And I'm currently an associate professor here at Oregon State University, and I'm also a principal investigator in the Linus Pauling Institute.
What is the Linus Pauling Institute, Alicia, for those of us who might not know?
So you may have heard of Linus Pauling. He is a two-time Nobel Prize winning scientist, and he did his undergraduate research here at Oregon State. And one of the things that he was really interested in at the end of his career was studying aging, and particularly healthy aging. And so he started this institute that's really looking at how micronutrients can play a role in regulating healthy aging. And so a lot of the researchers here in the Institute are looking at different types of micronutrients. I think you've all seen my paper about the acai supplementation. So that's kind of how I fit in. But we also just do basic healthy aging research as well. So it's a big mix of people that do a lot of different types of scientific approaches to look at these kinds of questions.
So, Alicia, one thing that we've regularly asked our guests is, how did you get into the world of fruit fly aging? Like, what was it that I guess got you into the sciences? What is it that got you hooked on it? And then already a question from the chat is, what even is an aging related disease? Because that seems like such a broad term that how does one then even figure out what in that field they want to study?
So I'll start, I guess, from how I kind of got into flies, and it was fairly random. So when I was in my senior year of undergrad, and at that time I was thinking I would do an MD, PhD, and but then I started doing my volunteer work in the emergency room, and I realized that I just did not have that personality working with patients and seeing them at their worst. That takes very special people, and I'm so glad that we have those people. So at that point, I decided I want to do a PhD. I can do biomedical research, and that way I can actually help lots and lots of people. And by the time I made that decision, I had missed a lot of the deadlines for applying for PhD programs. So I applied for a postbac at the National Institutes of Health called the Pre-IRTA program, Intramural Research Training Award. And I got accepted into that program. And it just so happened that the lab that I joined was a breast and ovarian cancer lab. And there was a piece of equipment that I needed to use for my project that was in another lab space. It was this small room. And there was one researcher in there that studied flies. That was where her fly setup was. And so I would just go in there and I would chat with her. And that was really my first kind of exposure to flies, like in the real world. As an undergrad, we did like a computer simulated, genetic crosses kind of thing. But I'd never really like seen research with a fly before. And when I got to graduate school, one of the faculty there was studying fruit flies as a system. and using it as a model to study kind of what are the beginning steps in cancer progression. So basically understanding what are the genes that regulate development, because in cancer, those are the genes that go off the rails. And so your cell, it has a job, but in cancer, it starts losing that job and goes back to like a more, immature state. And that was kind of the process we were studying. And I was really interested in cancer biology. So that's how I started in flies. And then all the amazing genetic tools that we have in flies, the fact that they're so similar to us. So like 75% of all human disease genes have an equivalent in the fly. And if you mess with that gene in the fly, you give them the fly version of that disease. So it's just such a powerful system. And so speaking of diseases, when I talked about aging-related diseases, kind of the big ones that come to mind, like Alzheimer's disease and Parkinson's disease, stroke and cancer. These are basically diseases that the symptoms begin after development, so after birth. A lot of times they'll start in adulthood. So when you think about like Alzheimer's disease onset is typically in the 60s, although there can be earlier and later onset. And so we're interested in understanding, why is this happening? And what are the different components that are contributing to those diseases? And then how do we just age naturally? What are the genes that are responsible for that process? And environmental factors, how do they play a role in that?
So how does one even begin a research question in aging like that? So you're having the fruit flies, right? They've got this similarity to us. And it still seems like it's a really, really broad field in terms of all the things that could go wrong with aging. Like, how did you decide what was your interest in the aging research? Like, how did you carve out, what? Because there's a lot of, even like the superfruits, the dietary addition that can slow aging, the disease progression. Like, how does one carve out like a good research question in such a broad field?
So that it really kind of goes with what you think is interesting and where you think you can fill in gaps. Now, I kind of stumbled into aging research. It was not my goal. So when I started my postdoc, my project was to study a gene called the P38 MAP kinase and its role in neuronal plasticity. So how do neurons, motor neurons, communicate with the muscle, and how do they make those connections with the muscle, and how does that change? And nobody had really studied this gene before in flies, and I was one of the first people to make a deletion of this gene, so I knocked it out of the genome. And one of the things I noticed about these flies is that they didn't live for very long, they didn't move very well, and they had a short lifespan. And so my postdoc advisor, he was like, as a control, you should show that overexpressing, so putting in too much P38, it doesn't do anything. And to my surprise, it extended lifespan by 37%. So these animals, so when you think about that, what does that mean for us in humans? So our average lifespan is about 78 years. This would be increasing it to 107. So it was just like a phenomenal amount of time that these animals are living. And so from there, I was like, I want to know how this is happening. And so, one of the things that we knew about what this gene does is it regulates oxidative stress. So this is where you have oxygen molecules that are missing electrons. And so what they want to do is, get their fix. And so they're going to steal stuff from anything they find in the cell. So that can be the DNA, it can be proteins, lipids, carbohydrates, you name it. And when they do this, they damage those molecules in the cell. So I wanted to know is what P38 is, the way that it's extending lifespan because it's regulating this oxidative stress, how the cell deals with oxidative stress. And I used genetic techniques, but then I thought, wouldn't it be fun to try an environmental, so a diet thing? And the way that started was I did my PhD and my postdoc at Emory University, which is where I met Belinda. And we used to go to lunch like every week to this amazing falafel place called Falafel King. And when we would go there, they always had that vitamin water, and they had the vitamin water XXX antioxidant. And that was my favorite one. And I got to thinking, I wonder if this really does have any benefits. Like, I'm drinking it because I like the taste, but does it really have any of these properties? And so originally I was going to test different vitamin waters, but the recipes are so different from flavor to flavor that I couldn't make really good comparisons. So then I was thinking about, well, what are the ingredients in this vitamin water? Of course, one of them is the acai berry. And so I got over-the-counter supplements that you can get from your local health food store or Amazon, wherever it is you want to order them from. And I started testing them. And I went in with the attitude that nothing was going to happen. And so I was so surprised when it worked.
So
So it,
why did you think nothing was going to happen? Because, like, right, we're always advertised of like, oh, antioxidants will make you live forever and they'll help you live longer. Like, how did you have that like almost skeptical, healthy mindset from the start of like, I have no expectations?
So I think one thing that's important for us to keep in mind is that vitamins and supplements are not regulated by the USDA. So they don't have to have any kind of like clinical proof of the claims that they make, like a drug that you would take for like a prescription drug or an over-the-counter drug like Tylenol or aspirin or something like that. And so with that, people may be making claims or they're over exaggerating claims. And, there's one thing that like a supplement may work, in a lab setting where you're taking this supplement and you're putting on an oxidizing agent and it, interacts with that. How that then translates to when it gets into your body has to go through your digestive tract. It's going to go through all that stomach acid. Is it still going to have any beneficial effects? And I kind of thought that when it went through the digestive tract, it would get basically destroyed and wouldn't have any benefits. So I was kind of going in with the idea that, we might see something, but I definitely didn't expect what we observed.
So something like the vitamin water, there's no need to actually evaluate what it's saying on there. It's just like, oh, we have this stuff on there and that helps, but there's nothing actually backing it up.
In a lot of cases.
Okay.
And I don't know, and a lot of times, the companies, they may run their own trials and get data from that. But as at that time, there were no scientific trials on, backing up the claims of these kinds of products. The specific products that I was testing are vitamin water. So, so since we didn't know, and in science, you'll also hear us talk about the null hypothesis. And what that means is that whatever you're doing doesn't do anything. And so usually that's a good way to start, is like nothing will change. And if something changes, and we have this, you'll have this referred to significance. And often what that means is that the changes that you're seeing are unlikely due to random chance. That it's due to whatever it is that you're changing about your system. And we typically use a cutoff of like, there's a 5% chance that what you're observing is random. That's typically the cutoff that we use. So that's kind of typically, the old school way of coming up with a hypothesis that it's not going to do anything. But typically, we actually start with something where we think there's going to be a good effect.
We had a question from the chat, Alicia. Did you use brand name or non-brand name, like vitamins and supplements, and do you think matter?
So we used, I guess, brand name. So there was a manufacturer. We actually named the manufacturer in the papers. So, each brand is potentially going to do things differently. And so when we did that study, because I was interested particularly in the acai berry, one of the things that I tried to do was that in all of the supplements, the concentrations that we tested were equalized to their purported amount of acai berry extract. But like some groups, they may have gotten freeze-dried acai berry, and that's what they used to make their extract. How they made their extract may be different. What it is dissolved in, because most of ours were pills that were like a powder. So like I opened up the capsule and I poured out the powder and I weighed it. And so, with that, that how they prepare their supplements could influence how effective they are. So there may be differences between, I guess, like off-brand, store brand, or generic brand versus like a company, like a well-established company's brand.
Gotcha. So even though if the doses of everything in there are the same, the processing may affect how something gets absorbed into the bloodstream, in this case through the gut. Uh, so,
Yeah.
so yeah. What did you find, Alicia?
Oh, so we found that so our P38 mutants, they have this basically accelerated aging. And we found that it rescued them. So what that means is that it restored their lifespan. This part's not in the paper, but it actually restored their locomotor functions.
Wow.
So, so it completely, made them so much better. It didn't get them all the way to 100%, but much, much improved. And then we also wanted to test environmental exposures. And so one of the compounds we tested is a herbicide called paraquat that is still commercially used today. It's banned, I think, about 60 countries across the world. It's banned in California.
Not the rest of the U.S.
Correct. So just in like California, not the rest of the U.S. And California tends to be ahead of the curve when it comes to banning what they consider to be toxic compounds. And paraquat has been linked to causing Parkinson's disease in humans.
Oh wow, that's weird. Then it wouldn't already be banned everywhere.
Well, there's currently a lawsuit. So if you Google paraquat, I believe that the lawsuit will come up first.
Gotcha. So it's, I guess, an even more biologically relevant thing than to test, right? For like in the experiment, because if it is already causing, manifesting disease states that are associated with aging in a young animal.
Yeah, and and people have tested paraquat exposure in all sorts of animal models, and it basically mimics Parkinson's disease in those animals, including flies. And so we know that it at least has the capability of causing Parkinson's disease in an animal model. It's really hard to test that in humans because it's kind of unethical to give people knowingly a drug that's going to cause, or a compound that's going to cause a disease. But we just kind of have this correlation data. So it was found that people that lived near where they did a lot of paraquat spraying in fields had a higher risk of developing Parkinson's disease than people who didn't. So that's how that association came about. So we thought, one of the things that paraquat does is it causes oxidative stress. So it causes the formation of oxygen radicals, and this is very damaging, particularly for our mitochondria. And so we thought, okay, this would be a good, really potent inducer of oxidative stress to test. And the compounds worked. So, which was also very exciting. We also tested hydrogen peroxide because that hydrogen peroxide in itself is a reactive oxygen species, a type of oxidative stress. And so, and that's, really simple to test. We know exactly what it does, whereas paraquat, we don't always fully know what it's doing. And the acai berry compounds also protected that. So, and I was like, well, is it because, we mix the hydrogen peroxide in the food at the same time we do the supplement? And I was like, well, maybe the hydrogen peroxide's being neutralized in the food. So it's never actually making it into the fly, and that's why they're doing better. So I exposed the flies to the hydrogen peroxide first, and then I fed them the antioxidants, and it worked. It protected them. And then I also, the reality is that, we're going to get exposed, and then we start having problems, and then we might take a supplement, right? Like that's kind of what we tested there, right? We don't tend to take medicines until after we start having symptoms.
Yeah.
But, we do, oftentimes try to take some things like vitamins. We take our calcium to improve our health, especially as we get older, right? And so we wanted to test whether or not if you could pre-treat. So if we fed them the antioxidants first and then exposed them, were they protected? And the answer was yes. So all of that has been really exciting. And it suggests that, at least for the flies, these supplements worked against multiple types of oxidative stress, either genetically caused by the loss of P38 or through these environmental exposures. And in all the cases, it made them better.
That's so. Is how do you interpret that to humans then? Does that mean that that vitamin water that was at Falafel King, is that worth getting, or is that still a leap away to be able to make that interpretation to humans?
So we're still at that leap. And so part of what we would need to do is what we call clinical trials. So, these products have been tested for some amount of safety. So they would go into a clinical trial where you would have people, just kind of like what we did with our flies, people that were on the antioxidant supplements, people that would be on a control supplement. In our case, we use ginger because it's going to have a lot of potentially amino acids and good, nutritional parts, but it's not really high in antioxidants. You may have a placebo control that's just sugar, but you would have these different supplements that people would be getting. And then you usually start with a small group to see if anything is beneficial. And then you would expand to more and more people. And there have been clinical trials using antioxidant supplements, but for a lot of these trials, they're for a disease. And the rules for a long time is that when we are doing clinical trials on people with a disease, it has to be kind of like the last ditch effort. And so when these trials were originally done, they were being performed with people who were already pretty far progressed, like in Parkinson's disease. So are the supplements going to reverse the damage? No. Mm-hmm. They probably would protect you from damage to begin with, or maybe slow down, the damage. So those studies didn't show any benefits, but again, by the time that the administration is happening, by the time the person is getting the supplement, it may be too late for them anyways. But people are doing these different kinds of clinical trials. Vitamin C, which is an antioxidant, actually was in a clinical trial for retinitis pigmentosa, which is an eye disorder. And that actually is effective, and it's currently one of the few treatments for that disorder. So it kind of just depends on, and it doesn't last forever. So it's not stopping the damage, but it helps slow down the progression of the disease.
I guess that's something similar that you saw in the flies too, right? It didn't keep them from aging, or even living past they didn't keep aging at the same rate. It just slowed down the rate, but you still had buildup of damage, presumably from other things as well.
Yes, exactly.
So how did that, Alicia, translate to your research today? And I know we had questions from the chat too about how you got into the sciences originally, but I think if you wouldn't mind, if we just travel down this rabbit hole of how that kind of study informed what your research is today, because that's a really, really cool finding, and it seems like there's so much to build upon.
Yeah, and that's an excellent question. And so, our results from the antioxidant studies was that it seems to only be a part of the story. So there's other things that P38 is doing besides regulating how the cell responds to this oxidative stress. And I wanted to know what that other thing was. And so part of that was by looking at what are the things that go wrong in aging. And one thing that happens is that you get problems with what we call protein homeostasis. So what this means is that proteins are basically the building blocks. These are, you have your DNA, you have your gene, it makes an RNA, which is basically the instructions. So you can I like to think of it as the IKEA manual. So your DNA is the IKEA manual written in Swedish. Right? Your RNA is the translation into whatever your language is. So for my case, it would be English. So that way I know how to put together my bookcase. And all the little pieces are the proteins that I'm going to need to put together to build my bookcase. So I now have a functional thing in my house, and it's this bookcase. So that's how your proteins are kind of working. And so It's really important. Your proteins, when they get made, they make a particular shape. So you almost think about it like gears in a clock. Like your gears need to be in a specific shape, or a key and a lock, or even just think about all the pieces that go into making a car. They all have to fit together in order to work. And so that shape is really important to their function. So proteins are the same way. And what can happen is that over time, these proteins get damaged, just like the parts of our car, and they get warped. We call this unfolding, or you might hear like denaturing. And the cell needs to either repair those proteins, or if it can't, it needs to throw them away or recycle them. So the cells actually really wants to recycle everything. And so that process gets disrupted in aging. So what happens is our cells aren't so good at recycling these damaged proteins anymore. And so I wanted to see whether or not that process was also disrupted in my P38 mutants, and whether or not increasing P38 protects against this damage to the proteins. And the answer was yes, it does.
So
And so,
one question we've got is how does one see something like that? So it's one of those, right, where it's like aging, you can look in, presumably, and say if it's dead or alive, or here we've talked about climbing assays to see how well they can climb up, but something like a gear and a protein being mismanaged, how can you tell when that something like that is happening?
so that's an excellent question as well. So there's a couple of different ways that you can do it. So for the flies, what we're doing is microscopy is one way. So we have a label on these damaged proteins. So when the protein is damaged and it needs to be targeted for recycling, it gets a tag added to it that says, hey, recycle me. And so we use what's called an antibody, but it's basically a tool that binds to that tag. And it glows when we have a laser light shined on it. And we can actually image that using a lovely fancy microscope called a confocal microscope. And so we can see these proteins clumping together, which is what we call aggregation or protein aggregates. You may have heard this term in the context of Alzheimer's disease. So how the A beta is the protein, one of the proteins that aggregates in Alzheimer's with these A beta plaques. This protein is all clumping together. And so we can see it that way. We can also use like biochemistry techniques. Again, we look for the number of proteins that have this tag, and are these proteins, when they start to clump together to form these aggregates, they become more dense. So we actually can separate them out by their density, and then we look to see, are there more proteins aggregating in our accelerated aging mutants. But if you really want to look at the structure, is that protein making the right shape? We have a couple of techniques for that. And one of them is X-ray crystallography, which is where you take this protein and you basically make crystals out of it. So it has to be kind of solid. And then you use X-rays to kind of, you remember probably seeing the picture, photograph 51 is a famous picture of DNA. So it's kind of like the same thing. You throw this X-ray at it and it scatters the light. And from that, you can determine the shape that the protein is making. A more recent advancement is this technique called cryo-EM, which is where you take your protein and you freeze it. And then you have a big fancy electron microscope that it's going to shoot electrons at your protein. And again, they scatter that light, they bounce off of it, and it will reconstruct the shape of that protein for you. It's absolutely amazing.
I mean, that it's beautiful. I know the community here really loves immunohistochemistry and microscopy, so a shout out to that. I think was really well received. Um, so yeah, that sounds like a very fruitful avenue to continue pursuing, right? What is your most exciting avenue right now, Alicia? So from that, because like with P38, you've got the behavioral side of things, the climbing, the aging, the physical ailments on the outside. You've got the breakdown on the inside. How do you determine, because you're trying to solve aging, right, as an overall concept, which avenue to go down and start pursuing first?
So one of the things that we've been doing is more proteomics. So what this is it allows us, it's a technique that allows us to ask, what are all the proteins that are present in your sample? So in our case, we can look at what are all the proteins in the brain, or what are all the proteins that are in the muscle of our flies. And we can have our flies under different conditions. So at different ages, we can give them the paraquat or the hydrogen peroxide. We can have P38 mutants, overexpression, combinations of all of that. And one of the things that we don't know is what are all the proteins that are aggregating with age.
Mm-hmm.
And so that's kind of our next step, is to figure out what those proteins are, and then how does that change with age? How does that change with exposure to oxidizing agents like the paraquat? And what are the commonalities? And can we find something that is kind of like that driving factor of aging? So, do all proteins aggregate? Do some of them aggregate and that's okay? It doesn't cause any problems? Or are there ones like specific proteins that are aggregating, and because they're doing that, it's toxic to the cell? And that's what's driving the process. So those are kind of questions that we're currently looking at. On our more diseased focused, one of the things that we're looking at is, why is it that you have a single gene with lots of different mutations? So we have a gene that we study that there's over 400 mutations reported in humans that cause over 19 different diseases. And these diseases are not related to each other. So some affect the motor and sensory neurons, some affect the certain fat deposits in our body. So not all of our fat deposits, but very specific ones. There are other mutations that cause one type of muscular dystrophy, and then other mutations that cause a different type of muscular dystrophy, where the muscle groups that are affected don't even overlap. And then, I know,
And it's
you have
all one gene.
One gene. Ones that cause cardiomyopathy, so problems with the heart, and that can be on its own or with a muscular dystrophy. And you can even have mutations that cause accelerated aging in humans. And all one gene. And these mutations don't cluster to a specific region in the gene. So it's not like all of the mutations that cause accelerated aging appear in this one area of the gene. So when we talk about the different regions of the gene, those different regions are going to become different parts of that protein. So we're going to affect the shape of that protein, just like we were talking about. And that could affect its function and what it's doing. And so, what's going on with this? And this is a gene that's present in like almost every single cell of our body. So why do some mutations cause this disease that affects these specific tissues and not others? So that's something that's really exciting that we've
Is
been
that
working on.
in flies too?
In flies, and we just got a grant to start testing this in organoids. So,
Ooh, okay. In so that it's really exciting, right, to have the corresponding in the fly as well. Can you make different results depending on where you mutate in the fly to, or can you even pop in the human copy and get something similar?
yeah, and that's what we've done is we've put in the human patient mutation. We engineered it to the equivalent in the fly gene. But you can definitely take that human gene and express it in the fly, and you can get similar results. We've been kind of focusing on just testing the fly gene because we know that all the things that it interacts with are present in the fly. And so the flies, as I mentioned, like 75%, human disease genes are found in flies, but flies are also just a lot simpler than us. And so they don't have every single thing that we have, but a lot of that basic parts of the pathway and all the basic elements of those processes are found in the flies. And that's a good place to start to understand what's going on. So that's why the flies are really handy. We can also do a lot of the genetics really easy. So we can ask, if we make this mutation that causes this disease, what other proteins are being affected by that? And if we now mess with that second protein or that second gene, how does that now affect the fly? Does it make them better? Does it make them worse? Does it depend on what we're looking at? And so basically we can figure out a lot of these kind of ground fundamental information about how these processes are working. And then we can take that into a human or a mouse system and then ask our questions. Because those kind of genetics things are much, much harder to do in those systems. And the other benefit of the fly is that it's a whole animal. And one of the things that's really cool about flies is that we have these genetic tools that we can use, that we can manipulate a gene in a specific tissue at a specific time. So I can ask what happens if I mess with this gene, and that can be, losing the function of the gene, or it could be gaining the function like our overexpression. What happens when I do that just in motor neurons? What happens if I do this just in this one muscle in the fly? So it allows us to really start understanding how different tissues are responding to mutations, and also trying to figure out what are the differences. So why is this tissue responding in this way and this other tissue isn't? So it's a really, really powerful tool.
One point that you brought up about, mixing the mutation with other genes and potential mutations and see how they interact. Someone asked if you've ever worked on the Methuselah mutant, so a deep cut on lifespan.
No, I haven't. That mutant has been kind of hard to get our hands on. So that was the last gene that Seymour Benzer was working on before he passed. So if you don't know who Seymour Benzer is, was one of the most amazing fly geneticists, and he was the guy that figured out that in his lab figured out that genes regulate behavior. And the way they did that was through circadian rhythm. So they had flies that they came out as an adults. So the flies have their life cycle where they're a little egg, comes an embryo, larval stage, and then they pupate. So kind of like the cocoon stage of butterflies, so you get caterpillar cocoon. And then out of that cocoon or the pupa case comes the adult fly. And this typically happens at specific times of the day. And so they were looking at mutants that cause the flies to come out to reach that final step at a different time of the day. And that was the period gene. And it was like the 200th fly that they tested was the one that did it. And I mean, this is just a back-breaking, labor-intensive assay that they did. But from there, they went on to show that genes play a role in courtship behavior. So you need to have a specific gene expressed in a specific part of the body, and that is what's going to allow the fly to perform courtship. And then also, he did a lot of work figuring out what are the genes that play a role in learning and memory. So there's a really nice biography called Time, Love, and Memory that's about him. So I highly recommend it. It was really good. Good read. But one of the things that he was somebody that went from when a lot of people started working on that topic, he would go to the next topic. So he was always looking into the future as to what was the next big problem that we needed to solve. And so aging was kind of the last thing that he was working on. So, but I think it would be really cool to test the Methuselah mutants. We've looked at the FOXO mutants. So FOXO is part of the insulin receptor pathway. And so that's a pathway that plays a role in aging across organisms. So flies, worms, mice. There's links that mutations or alleles in FOXO are connected to super longevity. So being like a centenarian or a super centenarian. So those are people that live to be 100 is a centenarian. And a super centenarian is someone who lives to be over 110. So people have been trying to
not the half human, half bull, right?
No, that's centaurs. Slightly different.
That's so again, you can, like you were saying, taking the fly equivalent and making the equivalent mutations, right, that you see in the patients, you presume you can do that for that gene as well, and then see what the results are. Again, showing that really cool, like shifting between the organisms and that conserved gene and just, figuring out how stuff works.
And those studies really, they went the other way. So it was animal models that first, or actually it was a yeast model that was the first to show an aging gene. And then it was a worm and then a fly. And it was in the worms that it was this insulin signaling pathway that was identified as playing a role in regulating aging. And so it was work from that then when when you're doing like a genomic study, so you're comparing the genomes of lots and lots of different people, between me and Belin, there are 100,000 differences in our DNA. And it's not a lot when we've got like 3 billion, bases in our DNA. So it's a really small fraction that's different between each and one of us. That's what makes us individuals. But that's a lot, 100,000. Like, how do you sift through which of those differences are important for, longevity or a disease or whatever trait it is that you're looking at? And so, when we look through that data, we can use the animal models to inform. And they're like, oh, well, we noticed that quite a few of the people that are centenarians have this allele or this mutation in FOXO, and that's been linked to aging in all these animal studies. This might be the thing that's driving aging in humans too. So we use the animals to inform the humans and the humans to inform the animals, and going back and forth that way. And as I mentioned, yeast is also used, and there are plant models as well, and all of the bacterial models. All of these models have value. They allow us to test different things in different ways, and all of that adds to our knowledge about what's going on.
So, and then another question, Alicia, is all these extra variables that you're adding on in that can influence aging. How does diet and like caloric restriction or addition, how does that affect aging? And then do you incorporate that into the model as well?
So currently, my lab, we don't do any kind of caloric restriction. But that is one thing that has been shown across organisms, like it's a uniform. Every organism so far that we've tested that you put on caloric restriction, like it extends lifespan, except there's two studies that were done with rhesus macaques, as a type of monkey. One study was performed with the University of Wisconsin, and the other study was with the NIA, National Institute on Aging. And with these two studies, they both did the caloric restriction, and they got different results. So in one study, the monkeys lived longer. The other study, they didn't, but the monkeys were overall healthier. So even if it didn't extend lifespan, it extended what we call health span. So health span is how long we live being healthy. And that's really the goal of a lot of our aging research, is not necessarily for us to live a really, really long time, but for us to live as long as possible, as healthy as possible. So that way we only are in a disease and suffering, the symptoms of illness for a short period of time at the very end of our lives. Right now, with modern medicine, is amazing. It's really extended our lifespan, and it has increased our ability to live with a chronic disease. But at the same time, we're living with a disease which may impair the quality of our life. We may not be able to do all the things we would really like to do. And so we want to change that. So we want it that people will be healthier for much longer.
Yeah, I guess that's the catch, right? It's getting someone to live longer, but the quality of life isn't better. Like you're still aging at the same rate, you just last longer now. Like that's not necessarily the ideal, right? You want that healthy span higher and maintain longer. Yeah, that's a
Yeah.
really makes it difficult, right? But it makes it like you have a regimented direction to go in. So, would you mind diving into a little bit about how you can translate some of this work? So I think that's also have been common questions from the community of like, you're working on flies, like we've talked about the genetic similarity, but where is like almost the actionable item that you would be able to do? And at what point is it passed on to someone else with like a different organismal expertise? And like how does that handoff almost happen? Like when do you decide like this is as far as I can take this in this organism and I need to go elsewhere?
So, really great question. And it really depends on where you're starting from. So, in my lab, one of the things we do is drug screening. So we're looking for compounds that improve the locomotor functions of our P38 mutants. And we're currently using FDA approved drug library. So these are drugs that have already been determined safe to use in humans. They're actually used for treatments of a variety of diseases. And so basically, we're looking for like off-label uses of these compounds. Could they be used for treating this type of symptom? And because these compounds have already been found to be safe in humans, if we find something in our flies, we could then, and we'd have to collaborate with people because I'm not a clinician, but we would collaborate with clinicians and other scientists that work with human patients. But we could take that drug and go into clinical trials. So it could be a pretty fast turnaround. If I'm starting with unknown compounds, then if we find something that's interesting, then that compound has to go through all those safety trials. So that delays it again. And for me, again, I'd have to partner with clinicians that would be interested in testing those compounds. Or I could publish my data and somebody else may see that and they decide to do the clinical trial. I may not be a part of it. So I believe in collaborative research. In fact, these days, I think most of us are doing collaborative research. It's really, how do we push our knowledge forward into ways that are going to be impactful and meaningful for us? And not just like our human health, but just like understanding how our biology works. And how do other organisms work and how are they impacted by different things that are happening in their environment, for example. So this is really important. And so we actually collaborate with, in one case, I have a collaborator that works with mouse models as well as human patient samples. I have another collaborator that also works with human cell culture models, and they actually work with humans. So they do clinical trials. So I have 3 different collaborators that all work with like humans or human cell culture lines or human patient samples. And so a lot of times, again, we can do these genetics in the fly that we can't necessarily do in these human-centric systems. And so I have this long-running collaboration with Victor Fondes at Emory University, who I've actually known since I was a graduate student. And we've been studying the effects of heavy metal toxicity. So particularly copper. Copper is a micronutrient. We have to have it. It's essential for us. But too little is really bad and too much is really bad. And in both cases, they can cause neurological or neurodegenerative diseases. And so we've been interested in trying to understand this relationship between copper and neurodegenerative disease. And so we work independently. We'll test these genes and we'll look at these interactions in the flies with and without copper, exposure from the environment. And sometimes we get the data back and I'm like, well, that's not what we thought was going to happen. And so I email Victor and I say, okay, look, this is what's happened. This is what we see. It's possible it could just be a fly thing. And then he'll email me back and he says, your data are beautiful. They match exactly what we see. And the benefit is that, they're using like a cell culture. Typically, they're using a cell culture model. So these are just, neuronal cells, or sometimes they might be like muscle cells that are grown in a dish. So they're not in the context of the body. They're not even in the context of the tissue they would normally belong in. Whereas in the fly, we have the whole animal. Mm-hmm. And we're able to show that what they're seeing in a dish actually translates to a living, intact organism. And so that's really powerful and shows that what we're seeing in these really almost artificial, isolated scenarios can be real, but we need to validate that. And so that can be something that's difficult, that, what we find in a dish, does that really apply when those cells are now a part of an organ or a tissue? When they're interacting with different types of cells, your heart has different types of cells in it. What happens?
And usually in a dish, it's a single monoculture, one type of cell. So for the folks at home, it's not necessarily representative of what's going on in the body. It's just more of a cleaner way that you can look at one particular cell type and how it's affected.
Yeah. And this is really useful to do that approach because, a lot of times all the stuff we're doing is so complicated. So we try to simplify it as much as possible so we can understand what's happening. And then we scale up and see, you're going from, a single cell type in a dish to then what's happening in an animal. And then maybe we go from that to what's happening in a person. So, they all feed into each other. They all have really important pieces or roles that they're playing in how we understand this puzzle that is our biology.
What has been your most fun experiment? So we had someone in chat wondering, just made the comment of, it sounds like we do a lot of crazy things to fruit flies, and if they could talk, oh, goodness gracious. What has been your favorite, or I guess most unique experiment, and then most interesting result that still gets you jazzed?
All right. So I think one of the more unique experiments that we do in my lab is called the soot assay. So this is where we take glass slides and we put them over a candle flame. And so when that candle flame is producing smoke, and that produces soot, so this is black material that coats the slides. And then we let the flies walk across those slides, and they leave behind their footprints, kind of like when we walk in the snow or walk in the sand on the beach. And so we can use this to ask, why do they not move so good? Are they coordinated? Are they walking normally? Is there something wrong that's happening with them? And so I think that's a really fun experiment that we do in the lab. We've also started doing traumatic brain injury, which is probably it's pretty traumatic, to be honest. But we have the flies. Flies don't have a skeleton. So when you have a traumatic brain injury, what's happening is that your brain is kind of floating in a liquid in your skull. So when you're hitting your head, your brain flies, one direction and hits the skull, and then it can bounce back and it hits the back or the other side of your skull. So you're getting trauma on both, different parts of your brain, different sides of your brain. So because flies don't have skeletons, what we do is we put them into a plastic tube and we stick this on a spring, and then we release the spring and it hits a surface. We don't want it to be too hard, but it's hard enough that when the flies kind of bounce, they hit the side of the vial, the plastic tube, and then they bounce back and hit the other side. And we can then look at, and we do this at mild enough that some of the flies will be knocked unconscious, just like what happens to some of us when we have traumatic brain injury. And then we can look at how do they recover from that injury. And one of the things that we're doing, as I mentioned, the Linus Pauling Institute is looking at micronutrients. So we're collaborating with Dr. Emily Ho, and she is an expert in some of these different micronutrients, in particular nutrients that come from broccoli and other cruciferous vegetables, as well as zinc. And so we're looking at how do those things play or interact with each other in traumatic brain injury. And we're in the very early stages of that, so we'll see what we get. If nothing interesting, then we may try some other supplements and see what happens there. But the idea is that we can use. You wouldn't think about this little fly being as a model for traumatic brain injury. So cool.
Is there a way that you can standardize, like when you're doing, because we've watched videos on stream before where it looks like it's the doorknob spring, right, that's keeping the door from closing, and you put them in the tube and lift them up and boom. Is that one where can you have it be predictable, where it's either a mild concussion, strong concussion, or essentially in that tube do you get everything and you somehow have to screen through it?
So we can change like that degree. And so one of the things that we've added to, we call it a HIT device, high impact trauma device, is that we've added a piece to it so that we always do the same angle. So that angle matters,
Gotcha.
the angle of the spring released. It also, what surface the tube hits really matters. If that surface is too hard, the flies will die. We don't want that to happen. If it's too soft, the flies won't have any trauma, because flies, they're used to being flown into things. They're pretty hardy little creatures. We don't tend to think about that, but they are. So you have to kind of get to, and that's one of the things like each lab has to do is, with the materials that they have, figure out what is the settings that give you reproducible results that mimic the type of brain injury that you want to see. And we can verify that by looking at, talked about immunohistochemistry. So we can look at whether or not there's any damage to the brain using that. We can also measure like inflammation and other markers of brain injury that are seen in people with traumatic brain injury. And so we can kind of confirm that what we're seeing is kind of equivalent to what's happening in a person.
That's really, really cool. Yeah, it makes total sense, right? The angle at which you'd pull it back, the thing that it hits, the intensity, and it's also interesting, as you pointed out, that some of these animals, like they are hardier than what we might expect. Like it's not just going to be any kind of surface being hit or any angle, and it just goes to show like how important it is to record every step that you go down.
Yeah.
Um,
Yeah. One of the things that we want to do is work with a physicist to measure the amount of force that is working in our setup. So that way, then we might even be able to automate it more so that it's like a consistent force that's being used rather than us with the spring that's kind
Talk
of being
about
held by.
collaboration, right? That you're able to pull on, like you've got the animal researchers on like the mammal side of things, and you got the physicists that you're pulling in. Like that's just like a it just goes to show how cool the research program is of what you're able to do and like the wide range of individuals that you can kind of pull in because it is such a big question of like aging.
We've just started a collaboration with a bioengineer. So they're over in the College of Engineering and they develop all of these amazing, we call them scaffolds. So, like these structures that you can get cells to um, grow on and then form like the structure of a tissue. And like, it's just amazing the stuff that these people from different disciplines do. And when you can get everybody together, and something that's a barrier is that we have all of our jargon that we use. I try to avoid jargon as much as possible. But that can be a barrier for collaboration. And so part of it is learning how to communicate with each other. Because sometimes I might use a terminology that means something very different. And so we're not on the same page because we're not thinking about things the same way. But when we can break down those barriers, it's amazing what we can do. So I think that's really cool.
One thing that I've always looked up to you, Alicia, is how well that you communicate with non-scientists as well. Like, and that
Oh,
kind of
thank you.
goes as you just said, like, it's not really jargon heavy. You're trying to account for the fact that everyone should be pulled in and engaged. So I know from you, our personal interactions, that this science communication is important to you. Would you just could you tell the party people like why it's important to you
Mm.
and how do you advocate for something like that, or even why do you advocate for something like that? Like you as a clearly powerful, successful scientist with a very awesome research program, why do you care that others know what you're doing?
I think that we need to share our science. I think we have such cool stuff, right? And I think we need more people coming into science from all different backgrounds because they're all going to have different perspectives and different ways of thinking. And so part of that is by sharing that excitement and what is all the cool stuff. And I think that the average person should be able to understand what we're doing and why we're doing it and why it's really important. If I say I work on fruit flies, a lot of people are like, what? And a common question I'll get is like, flies have brains? And I'm like, yes, they do. They're a lot smaller than ours. And we can dissect them out of their little heads and image them. It's like amazing what we can do. And so, um, I think it's important that people learn the value of all of these systems that we use and why they're important and how they help us understand ourselves. I also think that this helps fight science misinformation, which is very easy to spread. Sometimes that information, it sounds really reasonable, but it's not. And part of it is because they sometimes they use a lot of jargon, and we think, oh, that sounds smart. And I also think that jargon makes things inaccessible. It creates barriers, and I want to break down those barriers so that everybody can appreciate all of these amazing things that we're finding, and we can share all of that. So I'm really, yeah, passionate about science communication. I think it's important. I think as scientists, we aren't really trained in that, and we also get caught up in terminology. So I think one thing scientists are often not comfortable with saying like, we definitively find this, because there's always room for doubt. But in reality, we're like 99% sure. But sometimes the way we say it makes it sound like maybe we're 50% sure. And so, we need to use the language that people actually understand, that we're really confident in what we're talking about, and that, we don't sound so doubtful. And I also think that it's, really important for people then to know, how do we figure out what drug to use to treat somebody, or how do we come up with new therapies? A lot of people are really interested in the CRISPR, and we're using this for gene therapies now. Like, it's amazing. And it'd be great if people really could understand how that process works, because we're going to be using this in our bodies, or potentially our loved ones are going to be experiencing this. And so how does this work, and why is it safe? Well, how is this benefiting me, and really having a good understanding that this is, how it works, and why it's good for me. So I think all that's really important.
I love that. Someone made the comparison. It's a little bit like Shakespeare, that you learn that secondary language almost before you can actually even read the original book, and kind of how that science jargon is too. Is there anything that you would like to leave us with, I guess, on the research side of things now?
I think something that I think people should take away is that, as scientists, we're constantly amazed by what we find, that we don't know the answers going in. We start with a hypothesis. So we have an idea, what we think we're going to find. But sometimes we get the unexpected, and we have to shift our thoughts and be like, well, maybe I was wrong about that. So now I got to rethink things. And we're working on a big giant puzzle, and we're all working on different pieces of this puzzle, and we don't get to see the picture. So, we're all working on our own little project, but it all builds into this big thing. And so science is a community, and it all fits together in different ways. And so collaboration is really important, in teamwork. And I also think being a good science citizen is also really important. And I think these are things that we can all embody in our everyday lives.
Alicia, before we let you go, what do you define as a science citizen?
Well, I would say it's really anybody. I think it's anybody that's interested in science and wants to promote science. I think it's anybody that's just curious about the world around them and wants to learn more. I think just being curious. And so, everybody watching this is clearly a science citizen, because they're interested in all the different things that we've been talking about. And I think when we go out into the world and we talk to other people about science things that we've learned, and those people say, wow, that's really cool, now they've become science citizens. So we're
So
going to.
by those that you reach with your outreach, right, they can become like almost carrying the baton to the next individual, continue to get them excited about what you originally told that first person. It's not dependent on like the degree you have, right? It's just dependent on your passion and curiosity.
Exactly. And I'm going to share with your audience, because hey, I don't know if you've ever talked about um, Al Strudevant. So he was, so back early 1900s is when fruit flies were first started to be used as a genetic model organism. And this was done by Thomas Hunt Morgan's lab. They just thought, flies would be fun to work with. And in this lab is Al. He has his bachelor's degree. He's just working along, and he stays in this lab. And he's the guy that figured out that genes are on chromosomes, that there's sex linked genes, so like genes on the X chromosome. He also figured out that like genes are linear on the chromosome. So before, people didn't really understand like how our DNA worked. You gotta remember, when he was doing this, we didn't even know that DNA was the molecule of life. At that time, everybody kind of thought it was protein, because they didn't think DNA was complicated enough. But he did all of these groundbreaking experiments with a bachelor's. And he was so awesome that they made him faculty without ever getting his PhD. So anybody can do science. I do science with high school students. I do science with middle school students, and we actually just recently did some science with preschoolers.
Oh, wow.
And if you want, I'm happy to come back and talk about this paper, the black wanton bees. So this was a research study. It's published in, I think it's Biology Open Letters. that was all performed by 8-year-olds looking at bee behavior. And they wanted to know whether or not bees could solve puzzles. And so they designed their experiments, they performed their experiments, they did them in replicates. The paper was completely written by them, and they made all the figures, which are drawings, like they drew the little figures. It's really great. It's peer reviewed. It's real science that can be cited, and anybody can read it. Anybody can do science.
Alicia, seriously, thank you so much for your time tonight and for just, talking about your research and your passion, shining through and getting our community excited even more about the sciences. Awesome. Alicia, thank you again. And we can't wait to have you on another again sometime soon.
Science is for everyone, all backgrounds, all ages, all genders, and all walks of life deserve an education. Come be one of us.